Hi everyone! As you can see, the sun is out, it's 7 p.m. and I am being flooded with like natural light in the evening. And so April has just been a delight for me personally, because I am now getting up in the light and I go to bed. Well, it's a little bit dark then, but at least I come home in the light. It feels really good. I know that one of the people who is not joining our call today, but who is a very key collaborator with Tammy, Christine, she is fast asleep in Australia. So we are switching over time zones there. So welcome to everyone else that's watching from wherever you are around the world. I want to sort of just spend just a second to acknowledge the fact that these times are very Western Hemisphere sort of centric. And one of the things that we're going to send around during June, so during OHBM month, is a feedback form to ask you for your opinions on the demo calls and ask you if you change anything. And, you know, if you want to make a pitch for changing the times, I'm super, super open to coming up with better ways of doing that. But for now, thank you so much. Thank you for watching. And thank you to my incredible guests. I'm going to start with Ariel Rokem, who's going to talk about, sort of give us like a really big picture view of scientific Python. So Ariel, take it away.
Okay, yeah, so I thought I'd give here, I know a lot of the presentations that you've had in previous video chats were focused on specific projects and work within specific projects and how people can contribute to specific projects in sort of the open science ecosystem in neuroimaging. And I thought I'd present something that's slightly different, which is neuroimaging within the Python scientific ecosystem. And so the way I think of that is not as necessarily one project or a specific particular project with a team. Rather, it's something that I like to think of as a broader community of practice. So if you've never heard the term community of practice, it refers to people who share some kind of craft or profession. Imagine, for example, a group of people who are all interested in baking, right? So they're all doing things within that framework. Some of them may do baking as a profession. That's what they do all day. They bake bread for other people, and some might bake bread as part of what they're doing. Similarly, people who use Python in neuroimaging, there's a whole variety of a range of different involvements in that. There are some people who do that all day long, and that's what they do. They write Python code for neuroimaging, and they write open source software that others use. And others might use Python as part of their day-to-day research in some corner of their research, or as a hobby, as part of what they do for fun. So it's a very broad thing. So how did this all start? This, I think people, the Python language has existed since the early 90s, and it's a very nice language for a lot of different things, and it's open source, it's free, and I think pretty soon after people started using Python for a variety of things as a computer programming language, people realized that it can be used for science, and a whole bunch of scientists started writing different software that can do things that we want to do in science, like numerical computing and data visualization and things of that sort. And that community broadly is the SciPy community. So there's always a confusion, both in neuroimaging in Python and in scientific computing in Python, between the broader community and the library. There's a library called SciPy that we use that aggregates together a lot of numerical computing tools. And then there's the broader SciPy community, everybody that uses the community practice of people who use Python in scientific computing. And similarly, in neuroimaging in Python, people pretty rapidly after these tools came along started thinking, oh, maybe we could use this in neuroimaging. And I should mention Matthew Brett in particular, who sort of in the mid-aughts, together with Jared Millman and others as well, said, and in parallel also a group in France, Bertrand Thirion's group in France, started thinking, oh, maybe we should start moving into using Python. These are groups and people who were using, for example, MATLAB, proprietary systems before, and started writing a lot of code in Python. And started writing there. So there's a library called NiPy. You might, if you Google for that or look on GitHub, and I will give links afterwards, you can find this library of software called NiPy. But really, it became something much broader. It became a sort of this community of practice. And if you go to nypi.org, you'll find a list of different projects that fit within this community of practice. But if you're doing Python neuroimaging in Python yourself and you've developed other projects, we're always happy for people to add to that list other projects as well. And people show up, and it's actually fairly easy for people to add projects into that list. And we're really interested in broadening and expanding that list to encompass everything that people do, combining Python and neuroimaging.
So can I love hearing this sort of like big picture. And one of my real passions, which people who have watched this call have probably heard me say on literally every single one of them, is that open source code is able to kind of harness everybody. And you alluded to that the community is really broad, and it has lots of different people who are engaging for lots of different reasons in there. And so I wonder, what would be your pitch for someone who is just starting in grad school, and maybe they've taken maybe they've taken a couple of classes, maybe they haven't taken any programming classes at all, but they're turning up and they're about to do some neuroimaging. Would you encourage them to join the SciPy community and use Python for their analyses? And how would you sort of pitch to them that they should start doing that?
Yeah, I think part of my pitch is that the using Python in your research doesn't necessarily limit you to a certain range of tools that exist within that language. It's very, very expansive. Along the way, you'll be able to pick up tools from other research fields as well. The nice thing about using Python and this SciPy community, this kind of ecosystem of tools, is that it translates across many different domains. So in parallel to the community of practice of people who use Python in neuroimaging, there's an entire community of people using neuroimaging in astronomy, for example, or in biology, et cetera. And you'll be able to learn from the lessons that these other people have learned. The fact that this grew out of open source software made it so that, that the communication is very open across all these different projects. There's an annual conference, the SciPy conference. It's actually one of my favorite conferences to attend, although I don't go every year, and I wish I did. This conference takes place in Austin every year in the summer, so it's a fun place to go to and meet people. And one of the nice things about this conference is that it's very different from the discipline-specific conferences, where a lot of the time you go and you meet other people that are doing things that are very similar to what you do, and there might be this sense of competition with everyone around you. In this conference, everybody's your collaborator because everybody's working on different problems using similar tools. So we all want to help each other in some way. So it's a very pleasant community. It's also a community that's very welcoming to newcomers. So you imagine a graduate student just starting. There are many different ways to get help about this. All these projects and communities have mailing lists. There is a mailing list, neuroimaging in Python. I guess you can broadcast the link later on. The nice thing about this mailing list is that you can come and ask questions about pretty much anything in the intersection neuroimaging and Python. If you're somewhere there, you can come and ask questions about that. And there's a whole community of people who are really just happy to engage with newcomers and happy to welcome new people and help out to the degree that they can with the kinds of issues that people are coming across, including pointing out resources for how you might learn about specific things. And there are a ton of resources for people to learn. Documentation and tutorials and so on.
And what do you think is the biggest sort of misconception about SciPy?
I think the biggest misconception that I've encountered is that it's hard to install these tools. So this is something that I still hear sometimes from some of my colleagues. But isn't it really hard to install? And they point, some of my colleagues who work a lot with using MATLAB, they point to this and they say, it's so easy to install this thing. Until you point out to them that in some academic communities in countries where they don't have the budget to buy the MATLAB licenses, it's not really that easy to install. And it used to be hard to install. So when I was in grad school and I first got interested in this, you'd spend hours searching online for resources to explain to you how to install and compile all these tools. But it's become really, really straightforward to install all these tools in the recent years. it's a matter of a few minutes once you have the right order of operations. It really is a matter of a few minutes. I'll point to one resource in particular. There's something called Anaconda, which is a distribution of Python, and there for all the common platforms. You double click on something and it installs the scientific tools in Python onto your machine, and from there installing the neuroimaging specific things it's just another bunch of commands that you could type and install. It's actually really easy to install.
I completely agree. I did try to install some things on a Windows machine, and like everyone hates people that use Windows. A few years ago, before Anaconda, and Anaconda has changed my whole like ease of life in a pretty general way, exactly for that reason. Ariel, thank you so much. I'm going to keep an eye on the time, and I just don't want to sort of cut anyone else off. So I really appreciate the distinction between the SciPy library and the much broader SciPy community, and want to sort of really echo Ariel's point about it being really inclusive, and really everyone's pretty just excited to have anyone come along and care about writing some scientific code in Python. So bringing more people in, even if you don't feel that you're a sort of super high-tech developer, is actually like a key defining feature of the community. Not you won't be on your own for that. So thank you so much. I'm going to switch over. I hope I'm going to. Oh, I hide the participants. Wait, and then I gotta click this button, and it means I stop presenting Ariel. It's back to me. Hi, everyone. I'm going to switch over to Dora. Dora, it's lovely to see you. We have a few Skype calls. We've had a few, but it's really, I'm really looking forward to hearing about your experiences with iEEG. I'm going to let you pronounce that and define it, and BIDS. So welcome, and tell us about your project.
Yeah, thank you very much for giving me the opportunity to tell something about the iEEG BIDS. So iEEG BIDS stands for intracranial EEG BIDS. BIDS is a standard data structure for neuroimaging data, and in iEEG BIDS we're extending that to intracranial EEG data. This is a community-driven effort. This is not just, this is something that's driven by a lot of people who work with, a lot of great scientists who work with intracranial EEG data. And with a lot of help from Chris Holdgraf from the Berkeley Institute of Data Sciences, and Chris Gorolowski from the BIDS community. So we're trying to develop a standard data structure for intracranial EEG data. Intracranial EEG data include electrocardiography, stereo EEG, and deep brain stimulation recordings that are recorded in clinical settings, for example, in patients who suffer from epilepsy, or during tumor surgery, or patients with movement disorders who are undergoing deep brain surgery. And we sometimes have the opportunity to work with these patients and record some of these very unique data for scientific purposes. So these data are recorded in a very large amount of settings in the OR, in the intensive monitoring unit, and sometimes they're recorded on a large variety of clinical systems. Electrodes are implanted all over the brain in different formats, in different configurations. And in order to share these data, we needed to standardize the way in which people store that. And it needs to be simple. People need to be easily able to access it. It needs to be readable by clinicians, by technicians, by scientists. And it needs to allow for an easy integration with MRI data, because in order to know where these electrodes are, we need to co-register the electrodes on top of an MRI. So in order to do that, we extended the BID structure that was initially developed for fMRI data to iEEG. And the easiest way to kind of explain what it looks like is just by showing an example. So I would like to see whether I can share my screen. Let's see here. I think.
There's a green arrow on the left.
Yes, so I'm sharing my screen. So here we have a lot of examples for the that are in bit structure on GitHub. So this is just GitHub INCF BIDS examples. And here we are in the iEEG branch. We're in the iEEG branch where we have two examples currently for iEEG data in bit structure. And I'm going to just show this very simple example from a visual data set in bit structure. So you can see here in the root directory, there's information about the participants. And you can see that this is a tab separated format. So this is just a table where you can have an ID, an age, or a gender. You have a dataset description, which is a JSON file, which is also readable by machines as well as humans. You can just see it has a name, iEEG visual, a version, a license. So it's a very simple and human readable structure. So we go now in, for example, subject 1, the first session, and you can see here that this is where the anatomical data, which is in our general bits format, are stored. You can see there's a nifty file. And here you can see the intracranial EEG data. In this case, it was a set of ECOG recordings. And on this GitHub webpage, all the data sets are empty, but we can have a look at the metadata, for example, which include the electrode positions, name, XYZ coordinates. And these are all standard ways in which we store these data. There's a coordinate system such that we know how to interpret the data, such that we know that the electrode positions are stored with respect to the T1 weighted scan, such we know where the electrodes are. We can share these data with someone else, and they know if we share a list of electrodes with them, they know where they are. They can define their coordinates, look them up, see which brain areas they are in, and be able to interpret these data. Which is extremely useful. There's a standard list, for example, a standard way in which we can store events, onsets, durations, trials, links to stimulus files. So it's a very easy to read, well-defined structure. So this has been really, really fun to develop this. There's been a lot of people involved in this effort. And let's see, I can stop sharing my screen. Yes, I guess so. We've had a lot of contributions to the specifications, which are in a Google Doc and are shared with everybody. So everybody who can read the specifications, we've had a lot of input from, for example, a lot of different scientists who have looked at it, seen whether it's applicable to their data, whether they can store all the data that all the metadata that they would like to store. And we've had several extremely valuable contributions, for example, for people from M&E Python, Fieldtrap, iAlvis, to make sure that software packages can also work with these type of data.
Sorry, I'm going to flip over to me. So for me, I know we had a Guillaume talking about the MEG bids project in the first community call, and for me, I think the potential of having everyone just save their files in the same format and be able to interact with all of these different files is so exciting for being able to do reproducible research, so that other people can kind of check what they've done and they can maybe replicate in a different data set or something like that. But iEEG, the impression that I've always got is that the data sets are so heterogeneous because, you're getting this data from people who are already having an operation of some sort. So what do you get excited about when you think about the potentials of everyone using this? When everyone uses it, yeah.
Yeah, so one of the large potentials of everybody using a similar data structure is that we can start comparing and integrating experiments across multiple sites. What we've seen is that when we collect data, even between different sites, for example, between Seattle, NYU, Utrecht, Stanford, that our findings are generally extremely reproducible across sites. If we do a simple motor experiment that they did in Seattle, we had the exact same results in Utrecht. So so we generally do these studies in a relatively small number of patients with consistent electrode placement. But once we have a standard structure, we can actually, and everybody starts to share data in such a structure, we can start comparing brain areas, for example. We can start to know more about differences between sensory and motor cortex, difference across parietal frontal areas. We can really start, it really opens up an entirely new perspective for ECOG, where we can, for example, study age in a large group of subjects or development in a large group of subjects, because that's currently impossible, because it's just every site collects, 12 would be already a lot of, if we would collect 12 subjects a year, that would already be a lot. So it's having these data shared in a common format would open up a lot of new opportunities and a lot of new possibilities to study brain function.
I love it. I love this potential of sort of a whole bunch of new creative ideas for actually understanding the brain, rather than kind of just doing the same analyses that we've always done, because we're limited by what we collect. We're just running out of time, but do you have any, just really briefly, sort of requests from anyone that's watching? Is there any way in which people who are watching could get involved or could help out with our iEEG bids?
Yeah, so if you are interested, you can have a look at the specs. If you have some cool analysis code, if it would run on a BIDS dataset, that would be amazing, and we would be happy to incorporate that. So it's would be very excited to have any contributions from just people working with these data to make sure that they, that they are happy with what's going on, and that they're happy with the specifications. And analysis people, please make sure that your code runs on BIDS. There'll be a large community of people who can easily use it.
Thank you so much. Thank you, Dora. Just keeping one eye on the time, I could sort of continue to ask questions about this for so long. But Tammy, let me switch over to you. Thank you for joining us. You have a really, really cool idea that the Open Science Special Interest Group of OHBM are going to help to sort of pull off and make really special. Do you want to tell everyone about it?
Yeah, so we're super excited for this upcoming OHBM meeting in Singapore, that we're doing this really cool symposium. First of all, it's about naturalistic viewing paradigms, which I'm always excited about. The second really cool bit, I think, is that OHBM has allowed us to sort of pilot this new format. So it's actually going to be 7 speakers in one symposium. So each person only speaks for 8 minutes. So it's this idea that this is like super fast, lots of data, just a sort of different way to come at things. The third part, which is where you guys come in and the whole open science thing comes in, is that they're also allowing us to hold this sort of open science competition, where anybody in the whole entire community, doesn't matter if you're like an RA or what, a very senior investigator, can use this open source data set, the Healthy Brain Network, which you guys have talked about here before, and run some analyses that sort of highlight this movie watching data. And we're going to pick whichever sort of set of analyses looks the most innovative and kind of highlights the richness of this data. And that person gets to be a speaker in one of these speaker slots in our symposium. So it's this very cool combo of these very rich data sets that are open and available, and then putting it all together in this sort of, I don't know, new format. So we're very excited about it. We're a little nervous that it's all going to, go off smoothly, but so far it's looking really, really cool.
And I was looking at your the info that you sent along, and the deadline, I think, is in 1 month's time, right? I think it's the 25th of May. And
yeah,
do you want to tell people what they would need to submit?
So it's super simple. Basically, you would just submit a maximum of 10 slides. If you want to submit fewer to Christine, my co-chair, and I, that's fine too. But you basically just submit sort of a rough draft of what your talk would look like. Obviously, the data needs to be, and the analyses need, and the results need to be real. But you don't have to be super polished in like the, figures and all of that stuff at this stage. But yeah, it's a really tight timeline, which is both exciting and is freaking some people out. So we'll see what happens.
Sorry, me too. I think the movies are Despicable Me, I think, is one of them. And then do you remember what the other one is?
Yeah, so this is a developmental sample. This is the Healthy Brain Network that's spearheaded by Mike Millam and the folks out of the Child Mind Institute in New York. It's a wonderful data set. They've already released like a ridiculous amount of scans and subjects, very phenotypically rich. And then they have some resting state scans. And then there are 2 movie conditions. The long one is Despicable Me. So it's a 10-minute clip from Despicable Me. And then the shorter clip is about 3 minutes, a little bit over. And it's this animation called The Present. And it's sort of this very social cognition heavy, very emotionally evocative, you know, sort of pre-adolescent, adolescent kind of little short movie. So yeah, they're both meant to really drive these emotional responses, this engagement, right? This is not trying to be like, you know, middle of the ground. We picked something, you know, that Mike Millam wanted to go for these very, very specific responses. So those are the movies that people have to work with.
And they can download the data, and you've got the link, and we'll put that in the blog post right afterwards. And I suppose Ariel would probably recommend that they use some Python tools, maybe. But I wonder, do you have, I don't really want to kind of like bias the entries that you come up with, but do you have any like wild dreams? Like, are there any analyses that you would just be crazy excited about?
I mean, there's so many. I think that's the fun of the movie watching data, right? You can take task-based approaches, you can take resting state approaches, you can take weird hybrids in the middle. There's a big buzz right now about just sort of predicting, right? So we have this wonderful richness of phenotypic behavioral outcomes. So something that was predictive in that sense, I think would be really, really interesting. I also just think this age-based, right? This is a developmental sample. Like, when do you get that? So some basic age-based questions are still out there. And then I think one of the more recent sort of waves in the naturalistic viewing world is more data-derived, more data-driven ways of looking at the networks. So, you know, ICA or a PCA or something that actually lets the network structure that is being evoked by these very profound tasks come out, as opposed to us imposing our, you know, canonical network structures. I mean, that's just 3. I could go on for like, you know, a long time. There's so many interesting ways. And I would just love to see something that I haven't even thought of. So that's kind of what I am most interested in throwing this out to the community is there's so many ways to think about these data sets. Let's just, you know, see what people come up with.
I love it. I love it. And so everyone, you have lots of time. It's 4 weeks. You could do, you know, a couple of like big coding sprints over the weekends and get it done. And it will be really, really exciting to judge those. I think the Open Science SIG are going to take a look at these, and we'll be, you know, our sort of as unbiased as we possibly can. We'll just see what we get excited about.
Yes.
Tammy, I have a question that Elizabeth has copied over from Twitter for us, which is really fun. So thank you, everyone who's watching. This is not for the competition, because the competition is already on these 2 movies that have been collected. But do you have any recommendations for open movie stimuli?
Yes, actually, I happen to. So the way that my lab sort of started was with this movie that we created specifically to be used in sort of resting state approaches to functional connectivity, and specifically in kids, though it's been really useful actually in adults and in kids. So we spent a lot of time and money making this very sort of artistic, beautiful 7-minute animation called Inscapes. And it is, you know, totally, we own it. So it's open to the world and you don't have to copyright anything, which is great. And that one's can be down. I don't think I put the link for that one, but it's at headspacestudios.org. You can download that in uncompressed format. And the other great thing about it is it's nonverbal. So it's just music based. And so different groups on different continents are able to use it. And I think that that's like a really, really cool feature. So yes, that would be one. And I think, you know, long term, it would be just amazing if we had like a library of these like very well made very carefully constructed movies that are extremely evocative in certain ways. That was like our library of naturalistic paradigms that we could use as a community. Obviously, we're still going to need everything else as well, but having that as we sort of have our canonical Stroop and our canonical face task or whatever, I would love to see like some really cool movie versions of that.
I love it. There was actually a call for some project applications to the Alan Turing Institute in London, which is where I work. And one of those applications that I was giving a little bit of feedback on because it was for a friend of mine, Steve Eglin. He was thinking about trying to link up how the brain sees versus how a computer sees, and bringing together the computer vision communities and the fMRI or the EEG communities and pulling it together. And I just got super excited about the possibilities of all of these different sort of collaborations. And I think they really come from being able to share your data and being able to share your code. So thank you, all three of you, for kind of tying in that beautiful theme. We will get the blog post out, and so you'll be able to sort of see this recording, but also we'll put in all of the links that are related to those conversations that we just had. And please share the competition. 4 weeks, lots of time, anything goes. Just, come up with something fun, and you'll get to stand up on the stage and tell people at OHBM all about it. I'm really looking forward to seeing those. Okay, we will be back at the end of May. We're probably going to do, I think that one is going to be themed as a get excited about OHBM call, because we will be just a few weeks away from the annual meeting. But OHBM is a loving, open community all year long. So thank you to everyone who's here. Thank you to everyone who's watching. Thank you to our silent moderator and all-round superstar Elizabeth, who is always quietly doing everything in the background to make sure all of this runs. And have a lovely day.
Thank you.